I would like to thank the organizers for inviting me to give the talk on advancement in Leishmania in vaccine development, role in global elimination of Leishmaniasis. My name is Hira Nakhasi, and I work for U.S. and Food and Drug Administration. My contributions are an informal communication, represent my own best judgment. These comments do not bind or obligate FDA. Overview of the presentation. My presentation is as follows. I will discuss first the leishmaniasis and its global impact, vaccine as a tool for achieving control and elimination, various vaccination approaches which have been described in the literature. Then I will talk about the preclinical safety, immunogenicity, and efficacy of genetically modified live attenuated vaccine candidate, L major central parasite, in various animal models, which this vaccine was developed in my laboratory. And then I will discuss some of the mechanism of the immunity of Leishmania major central parasite. Lastly, I will discuss the status of this vaccine and Leishmania skin test development, focus on the cGMP manufacturing of the vaccine, toxicity studies, and the future clinical trials. Overall, global impact of leishmaniasis is felt in both subtropical and tropical regions of the world. It comes into various flavors, visceral disease, cutaneous disease, and mucocutaneous forms of this leishman disease. It is endemic in 88 countries. Approximately 1.2 billion people are considered at risk. Every year, 0.5 to 1 million new cases appear, and there are more than 12 million people are infected each year throughout the world. Approximately more than 50,000 deaths annually are reported. And in addition to that, because of the environmental changes, it is expanding its footprint into regions where it did not exist before. For example of such is that WHO declared US as an endemic country since the autochthonous cases of cutaneous leishmaniasis were observed in the southern part of the United States. And finally, there is no vaccine available for this disease. The life cycle of Leishmania parasite is in two forms. One, it exists in the sandfly invertebrate host, and the other one in the vertebrate host, which includes human beings. In the sandfly, the parasite form is called the promastigotes. They go under different degree of differentiation, eventually become called metacyclics, which are infectious, and that stage, then sandfly injects into the human or vertebrate host to seek a blood meal from that animal or humans, and injects these metacyclics into the host. In the host, these metacyclics are taken up by macrophages and other types of mononuclear phagocytic cells. They are differentiated into amastigotes. These amastigotes then multiply in various types of cells and infect other tissues, and then the cycle is completed by when the sandfly comes to take a blood meal from the vertebrate host. Now, several years ago, a model was set up to see how a vaccine can eliminate this disease. In this model, they figured out if there was a vaccine which is 50% efficacious, what will it take to achieve the WHO recommendation target of elimination of 1 in 10,000? And to achieve that, if there was a 50% efficacious vaccine, it will take 8 to 10 years from the current status of 5 to 10 cases per 10,000, which includes the reservoirs of Leishmania, which are VL cases, PKDL cases, or asymptomatic cases. There have been many approaches for the vaccination against leishmaniasis. For example, people have used recombinant proteins and peptides. They have developed DNA vaccines expressing many of these proteins. They have used the whole parasites inactivated through heat inactivation, autoclave, chemical treatment, and also use live attenuated by making them chemically attenuated or radiation and culture. Unfortunately, none of these approaches so far produce a viable vaccine except for leishmanization. What is leishmanization? Leishmanization method was experienced in cutaneous leishmaniasis in former Soviet Union, Israel, and Middle East, where a small virulent parasite, small dose of virulent parasite was injected into people, which caused a small lesion, and after the lesion was recovered, and people developed a lifelong protection. Similarly, it was also shown in the literature that people who were exposed to cutaneous leishmaniasis, when they moved and recovered and moved into the areas where visceral disease was endemic, they were protected against the visceral disease. So then this brought us the idea of that can a genetically modified parasite, which does not cause disease, can do the same kind of cause same kind of protection. So what are the advantages of the genetically modified live attenuated vaccines? Because, for example, it mimics the natural course of infection, provides complete array of antigens, it has a controlled persistent without causing disease, and also there are genetically modified mutations which are well characterized, unlike other methods of attenuation. We, several years ago, identified a gene called centrin gene, which is a calcium-binding cytoskeleton protein found in all eukaryotes, most localized in the basal body of the lower eukaryotes and the centrosomes of the higher eukaryotes. This gene protein is important for the duplication and segregation of centrosomes or basal bodies. This was we and others had defined its function. So, using that as a centrin gene, we deleted the centrin gene in the L major L major parasites, and using CRISPR-Cas technology. And to give you a little bit background, centrin has 5 genes in the parasite, and centrin 1, which we had previously shown, that is important for this differentiation into A mastogotes, and whereas the other 4 are not. And when we deleted the centrin gene, when we did the whole genome sequence, only the centrin 1 gene was deleted. And when we looked at the growth both in the promastogotes and the amastogotes in vitro, the apromastogotes grew, whereas amastogotes did not. In the infected macrophages did not grow. When these parasites were injected in the mice, it didn't cause any lesions. However, when this centrin gene was added back to this deleted parasite, it regained the infectivity, suggesting that centrin is very important for the infectivity. We looked at the preclinical safety of L major vaccine in several animal models, which included the immunodepleted or immunocompromised animals, both the mice and the hamster, and showed that there was a lack of lesion development or lack of disease development. They were safe in immunocompromised and immunosuppressed animals. In addition to that, when these infected animals were fed on to a sandfly, and when the parasites were taken up by the sandfly, but they did not survive after a few days, suggesting that it can be environmentally safe as well. Then we looked at the immunogenicity of this vaccine candidate in various animal models, mice, hamster, as well as the PBMCs from non-endemic human controls. What the results showed that this vaccine induced a very strong Th1 response, both in all the models, and it suppresses the Th2. I don't want to go to details of this data here because it's already published in various journals. So we next tested the preclinical efficacy of the L-major-centrin in both these against the cutaneous disease as well as the visceral disease. The visceral disease was tested in the hamster model because it is a gold standard for human disease, and showed that when these immunized animals were challenged with the sandfly-mediated Leishmania donovani, which causes the visceral disease, it protected vaccinated animals. There was no mortality, no weight loss, no splenomegaly, no hepatomegaly compared to the non-immunized mice, which succumbed to this infection. Whereas in the case of in the mice model, we demonstrated that when immunized animals, when challenged with the cutaneous L-major parasites through sandfly challenge, they did not develop any lesions. Next, we tested this vaccine in the field against the natural infection in dogs. The dogs were immunized for over a period of 3 years. Every year they were immunized and then taken to the field during the season when the sandflies carry the infection. And this infection carried by these parasites is L. Infantum, which causes the visceral disease in the dogs, and showed that after 4 years of exposure and these to vaccinated dogs, they developed a very strong Th1 response, and their efficacy, they did not develop disease, i.e. the efficacy of this vaccine was greater than 75% compared to the non-immunized dogs. Now, as I mentioned earlier, we wanted to understand what is the mechanism of immunity of these L-major parasites. How are they developing such a protective immune response? So we tested several hypotheses. One was, how is it changing the early alteration in immune pathway? Second, we wanted to see what are the metabolic changes in the immune cells which results into the alteration of the immune system there, as well as what's the mechanism by which these parasites cause a long-term protection, and we found that it causes the modulation of immune landscape in bone marrow. In the interest of time, I will be only talking about the early alteration in the immune system. In this study, what we use is using transcriptomic analysis in which the RNA from air of wild type and vaccinated animals was analyzed, and after 2, 7, and 21 days of post-infection were hybridized to genes which were on the nanostring platform. What we observed, there was significant up and down regulated genes in the vaccinated air compared to non-immunized or wild type infection. Intriguingly, among all those pathways, what we found out that the transcripts associated with the type 1 or interferon pathway were enriched, including the IRF7. Now, what is IRF7? IRF7 is a transcription factor for the type 1 interferons. So when we immunize animals with L-major-centrin or infected with L-major wild type and looked at the expression of this IRF7 in the ears of these animals, what we found out that in both wild type and centrin immunized animals, the expression of the IRF7 increased between 2 to 7 days. However, in the vaccine, there was a sharp decline after 7 days, whereas it continued to stay up in the wild-type infection. These IRF7, as I mentioned, is important for the expression of the type 1 interferon alpha-1 and alpha and beta. And so we looked at the expression of the type 1 interferon in cytokines in the serum of these animals and found out that it follows exactly the expression of IRF7, i.e. it is up early on and then it goes down in the case of the vaccinated animals, whereas in the case of the wild-type infected animals, it stays up. This expression of this IRF type 1 interferon leads to the Th2 response in these animals, whereas in the case of the vaccinated, the suppression of Th2 results in the induction of Th1. Now, to prove this point that IRF7 is an important player for this responsible for the early events, which we had described in a minute before, that we tested the same, our hypothesis in IRF7 knockout mice, where we infected or immunized these IRF7 mice. We saw exactly the same. There was a lack of IRF7 type 1 interferons. And when these animals were either immunized or challenged, the immunized animals, which were then later on challenged with the wild type infection, controlled the infection similar to what immunized animals did. In fact, certain cases it did better. So suggesting that lack of suggesting that L major centrin modulates the expression of IRF7, modulates the type 1 interferons, and therefore results in the induction of Th1 response and causes the strong protective immunity. Lastly, I would like to talk about the manufacture of the status of the manufacturing of the L major vaccine under GMP conditions, as well as our efforts to develop the Leishmania skin test as a surrogate marker of immunogenicity for field studies. In collaboration with our partners, Genova Biopharmaceuticals, they were able to manufacture under GMP condition in a bioreactor large-scale production of this vaccine. And this vaccine is then, when tested in the preclinical studies, behaved exactly both with the safety, immunogenicity, and efficacy as the lab-grown parasite, suggesting that the GMP production did not alter any characteristics of this parasite. Using this vaccine parasite, we then tested its adverse events, if any, in the rat model. And when we looked at the toxicological studies using several parameters, we did not observe any adverse events and found a strong Th1 response in the rat model. Again, suggesting that it is safe. The GMP grade material is safe and doesn't cause any adverse reaction. Next, we developed this Leishmanian antigen, which was made from Leishmania donovani, which was a well-characterized parasite, and made once they were grown and then subjected to freeze-thaw, either freeze-thaw or lyophilization methods, and then the antigens which were produced through this process were tested against the tested for their DTH, delayed type of humoral response, delayed type response in vaccinated and leishmanized animals. In both the what we did was we immunized hamsters or with a vaccine, or gave a low dose of wild type infection after 17 weeks of treatment. When the parasite had gone from the animal, we then injected this Leishmanian antigen into the air of these mice, hamsters, and we saw a strong DTH response within 24 to 48 hours, suggesting that this preparation, our LST, Leishmania skin test, can behave as a surrogate marker in the vaccine and efficacy trials. So the current status, as I mentioned, is that with regard to L major centrin, we are now at a stage where we are developing pre-IND and IND meetings, and have consultation with FDA and US FDA, and we are all with the regulatory authorities in different countries, and also we have planning for the phase 1 studies in non-endemic and as well as endemic countries. So take-home message from my talk is that genetically modified Leishmania parasites are safe, immunogenic, and efficacious against the natural infection of both CL and VL. It's a safe alternative to leishmanization. We have discovered some new pathways by which this vaccine induces immunity, achieved a large-scale preparation of the vaccine, and it has no adverse event. And also, as we have developed a Leishmania skin test as a surrogate marker, and this vaccine is ready for testing in the clinical trials. Lastly, I would like to acknowledge my team in the US FDA, as well as my partners in this collaboration, Ohio State University, NIAID, Genova, INTOX, Pasteur Institute, Nagasaki University, and my clinical colleagues. The funding of this was provided by the Global Health Initiative Technology, National Institute of Health, Wellcome Trust, as well as US Food and Drug Administration. Thank you.